Welcome to Ask the Expert. Ask the Expert is really just a casual way it's a brief, informative, engaging, and lively discussion about cutting edge science and advancements in type 1 diabetes research and related interdisciplinary research. So we'll have a few slides and then we will have a discussion about the scientists' work. And today we're very lucky to have with us Dr. Alma-Martina Cepika. She's an MD, PhD from Stanford. Title of her talk today is Antigen-Specific Type 1 Regulatory T Cells, Mechanisms of Function and Therapeutic Use. So Dr. Cepika is coming from Stanford to speak with us today. She's an immunologist and she has an extensive background in translational research, autoimmunity, autoinflammation, and human systems immunology, all things sort of near and dear to the T1D research world. Her goal is to understanding the mechanisms governing immunological tolerance and leverage this knowledge towards new cures. So Dr. Cepika received her MD and her PhD in immunology from the University of Zagreb School of Medicine in Croatia. And while there, she was focusing on the immunomonitoring of patients with lupus, identifying how circulating DNA levels changed with therapy. So for her postdoc training, she joined the lab of Dr. Virginia Pascal at Baylor Institute for Immunology Research in Dallas, Texas, where she studied the drivers of autoimmune inflammation in systemic juvenile idiopathic arthritis. And she developed a multidimensional in vitro stimulation assay to evaluate immune responses of blood leukocytes in pediatric patients with this disease. So in combination with integrated bioinformatics analysis, this approach identified aberrant cellular responses, transcriptional pathways, and genes that shed new light on immune dysregulation in systemic juvenile idiopathic arthritis. Sounds like a very interesting response and one that could possibly and potentially be applied to an autoimmune disease like type 1 diabetes. And right now, Dr. Cepika is an instructor of the Pediatric Division of Stem Cell Biology and Regenerative Medicine at Stanford University School of Medicine. And there she's working to uncover the molecular mechanisms governing type 1 regulatory T cells, TR1, differentiation and function, and to use this knowledge to design TR1 cell-based therapies for hematopoietic stem cell transplantation, cancer immunotherapy, and autoimmunity. And she's funded by the National Blood Foundation, Stanford Spark, and Stanford Translational Research and Applied Medicine programs. Thank you so much for joining us. You have a wealth of experience. And I was really, we reached out to you, because we saw that you had this great Science Translational Medicine paper, allo-antigen-specific type 1 regulatory T cells suppressed through CTLA-4 and PD-1 pathways and persist long-term in patients. And we're hoping you can share some of that work with us today. And, again, it's a pleasure to have you.
It's a pleasure to be here. And thank you very much for inviting me. And thank you to everyone who managed to join. Let me start sharing my slides. Okay, so let's dive right in it. So T cells develop from hematopoietic stem cells in the bone marrow, but they do not finish the differentiation there. They actually, from the bone marrow, they travel to the thymus, where they rearrange their incredibly diverse T cell receptor genes to form an antigen receptor, which we call T cell receptor, or the TCR. From the thymus, T cells emerge as two main subtypes, which are the, let me see if I can use a pointer. Do you see my,
I do see the cursor. Okay. Yeah, that's
okay,
good.
Great. So, sorry, the T cells emerge from the thymus as either CD8 T cells or cytotoxic T cells, or CD4 T cells, also known as helper T cells. And these helper T cells actually have two subpopulations. First is the mature naive CD4 T cells. And the second are the CD4 T cells that express transcription factor FOXP3, which are better known as thymic or natural FOXP3 regulatory T cells, or Tregs. The mature naive CD4 T cells, however, do not finish their differentiation in the thymus. They actually undergo further development in the periphery once they're exposed to antigens. If they're exposed to antigens in an inflammatory environment, they differentiate into different subsets of effector CD4 T cells. But if they encounter a persistent antigen in a tolerogenic environment, they become type 1 regulatory T cells or TR1 cells. Both CD8 and CD4 effector T cells are instrumental for our body's defense against pathogens and tumors, and regulatory T cells basically act as a brake on the effector T cell responses, and as such, they're instrumental for the defense against autoimmunity, allergy, and they also are important for reducing transplant tolerance. TR1 cells are a less well-studied subtype of regulatory T cells, but we do know quite a bit about them. We can identify them by their co-expression of surface CD49B and LIKE3. We know that they produce very high levels of immunosuppressive cytokine IL-10 and express low or no levels of XP3. In terms of their in vitro and in vivo function, they are maybe best known by their ability to induce tolerance after allogenic transplantation, whether that's an organ transplantation such as eyelid or pancreatic transplants, or in the context of hematopoietic stem cell transplantation. And allogenic hematopoietic stem cell transplantation, or allo-HSCT, is actually our current focus in the Roncarol lab at Stanford, because the allo-HSCT is a very important and life-saving treatment for many, many patients with high-risk leukemias and also some patients with non-malignant autoimmune disorders. But because of the HLA mismatch between the donor and the host, the donor's T cells can sometimes attack healthy patient tissues and cause graft versus host disease, or GVHD, which is potentially a lethal inflammatory condition. And the risk of GVHD actually deters many hematologists from using this potentially life-saving therapy. So we really need to find a way to improve the outcomes of allo-HSCT without relying on general immunosuppressive drugs, which are actually harmful by themselves. And to do this, we designed a therapy that we call T-allo10 cells, which we make from allo-HSCT donor CD4 T cells, which are co-cultured for 10 days with patients' tolerogenic dendritic cells. So during this co-culture, these patients' dendritic cells instruct an autoreactive portion of the donor T cells to clonally expand and differentiate into TR1 cells. These TR1 cells have a constricted TCR repertoire, and they are alloantigen specific, which means functionally that they suppress alloreactive effector T cells, which can cause graft versus host disease in vivo, but they do not suppress T cells that have other TCR receptor specificities, such as the T cells that protect the patient against pathogens or tumors. And as such, the TR1 cells as a cell therapy can have a unique advantage over non-specific immunosuppressive drugs, such as corticosteroid or cytotoxic drugs that are used to suppress these allogenic immune responses post-transplant. The allo10 cells are already in the clinic. They are being tested for safety in 2 ongoing phase 1/1b clinical trials in patients with leukemias that are treated with allo-HSCT or with haploidentical transplant. And we actually just recently published the immunomonitoring result from the first trial here in this manuscript, where we followed up the patients receiving T-allo10 cells and allo-HSCT in 19 different time points post-treatment. And we also performed a detailed in vitro characterization of T-allo10 cell product by flow cytometry, RNA-seq, TCR-seq, and we also characterized their functional properties. Just to briefly summarize this paper, because it's published, so we showed that the T-allo10 cells contain around 10% of TR1 cells, And these tier 1 cells have a unique transcriptional signature. And through this transcriptional signature, we identified high levels of expression of CTLA4 and PD-1 and PD-L1, which are molecules that we knew they are involved in suppression of FOXP3 regulatory T cells, but they have not been described so far in tier 1 cells. So using suppression assays, we demonstrated that tier 1 cells indeed suppressed through CTLA4 and PD-1, PD-L1 pathway, in addition to suppression through the IL-10 pathway, which was known from before. So the T cell receptor also serves as a natural barcode of each T cell, because the T cells that share the same T cell receptor are thought to have the same clonal origin. And we can use this information to track these cells after infusion in the patients. What you can see here is that because of the tier 1 cells from the TL10 cell product are clonally expanded and are antigen specific, they have this very, very high enrichment of the top 100 most frequent TCR clonotypes, which are virtually not present in the total CD4 T cells from the donor, or present at very, very low level in the non-tier 1 cell component of the TL10 cells. And we use these top 100 clonotypes to figure out whether there are tier 1 cells in the patient whole blood in all the 19 time points post-TL10 cell infusion and post-transplant. And also we investigated whether these TL10 tier 1 clonotypes can be found in ex vivo isolated circulating patient tier 1 cells. And indeed, that's what we found. We found that the TCR clonotypes from infused TL10 tier 1 product are found in patient peripheral blood, and also in isolated circulating tier 1 cell from the patient, here shown at day 300 post-transplant. So this long-term survival of tier 1 cells post-infusion is very important finding that will be crucial for determining their clinical efficacy once we progress to hopefully phase 2 and phase 3 clinical trials. And since we know from a lot of CAR T cell work that if the infused T cells are not found in the peripheral blood, there is clinical efficacy is lost. And we also hope that this manuscript that really show how we can make and characterize tier 1 cells will encourage other investigators to explore tier 1 cells as a therapeutic strategy for other clinical indications, such as organ transplant or allergy or autoimmunity, since tier 1 cells can be relatively easily induced in vitro. So with that, I would like to briefly introduce you to the wonderful team of scientists I have a privilege to work with. So this is Maria Grazia Roncarolo, who discovered tier 1 cells more than 30 years ago, and she is a sponsor of these 2 clinical trials. These are the lead clinical teams, and these are the research folks with me who are with me in the lab. This is our last pre-pandemic photo on a conference. So with that, I will stop, and I'm happy to take any questions.
Thank you so much. That was really nice. I did have a question. So this seems, it might be, a great way to interface with eyelid transplantation. Can you talk a little bit about how that might work hypothetically?
So yes, so in theory, we can, in organ transplantation, we can envision to take the CD4 T cells from the patient and then generate these tolerogenic dendritic cells from the donor monocytes and do the opposite. So instead of let me reshare, maybe it's going to be easier to explain. So instead of taking the donor T cells and making the donor T cells tolerant to the patient, we will take the patient T cells and making them tolerant to the donor antigens. And because the dendritic cells, the alloantigen is nothing but normal proteins that are expressed by dendritic cells that are presented in the context of an allogenic class 2 or class 1 receptors. So this concept has been shown that it already works, not for diabetes, but it has been shown to work for organ transplant. So the same cells, which were not called TLO10, the investigators named them a little bit differently, but basically the process works. So we can take the dendritic cells from the donor and make TR1 cells from the patient, and then they will be tolerant to the donor antigens.
And, you showed that this process works, up to 300 days, right? So that's almost a year. So that's pretty helpful if you want to talk about, keeping an eyelid in place and not rejecting it.
That's correct. So we know that there has been several studies, and this was done by Maria Grazia Roncorolo lab while she was still in Italy. So there were several studies that showed that the TR1 cells can induce long-term tolerance to eyelid transplant in vivo murine models. And what we know from the earlier pilot clinical trial of TR1-like cells, so this was not the TLO10 product, so it was IL-10 anergized cells that were administered to patients, adult patients undergoing haploidentical transplantation. And I think this is published here in this paper. So this paper also shows a long-term immunomonitoring of these 5 patients that engrafted and achieved long-term tolerance. And I think the data in the paper is 5 years post-transplant. Now, they didn't do TCR sequencing, they just did phenotyping of TR1 cells by 49B and LAG3 co-expression in the memory T cell compartment, but they showed that actually the percentage of TR1 cells in the peripheral blood increases the longer it is post-transplant. So the highest level was 5 years post-transplant. Even if we don't know if these are the same TR1 cells that were infused, it seems that they indeed can be found long-term in vivo.
And so how, I guess, do you know of any labs that are currently interested in that particular approach, or, kind of like, I know you guys are more into, you're the graft versus host, but is there any lab specifically that's doing that type of work that, that we've just discussed in, exploring eyelid transplantation in humans and using this system?
I don't think so. At least not to my knowledge. I think that Maria Grazia Roncorolo always wants to kind of continue with the research into diabetes. I know that she's an active member of JDRF, so it's still somewhere in the back of my mind, or back of her mind, that she wants to restart this, and maybe she actually continues with it now. But the postdoc who published this work in diabetes, her former postdoc, now has a lab in Europe, and I don't know if he's continuing with diabetes research. It's a good question, but it certainly has a good basis to continue with generating TR1 cells to induce tolerance to eyelid transplants.
Yeah, I mean, I'm surprised actually, because there's such a push with Vertex and ViaCyte all trying to get good results from eyelid transplants. What would be the risk, I guess, of using this, layering this kind of therapy on top of an eyelid transplant in a human, and ending up with like teratomas? I mean, is that something that might arise? Because
I in my mind, no. So we have from the first clinical trial, we have 4 patients completed by now. The first 3 patients were described in the paper, and the clinical outcomes are so far very promising. So these are patients with very high risk leukemias. Some of them failed, the first transplant. Some of them failed CAR T cell therapy, and they're all alive and doing well. Because tier 1 cells are generated to be antigen specific, they should not, in theory, and what we also confirmed in vitro, they should not impair any responses, T cell responses that are protective. So they shouldn't impair protective immunity against pathogens. They shouldn't impair protective immunity against tumors. Now, in the context of the only thing that it could possibly be modified for organ transplant is to get rid of these polyclonal effector T cells from the TLO10 product. So in the TLO10 cells, you see the tier 1 cells are relatively minor fractions. So most of it is polyclonal CD4 T cells. And for transplant, this is actually okay because these patients receive much higher dose of polyclonal CD4 T cells with the graft. And these polyclonal CD4 T cells are actually important for mediating anti-leukemic effect. But this is maybe something that should be modified for organ transplantation, because infusing a lot of polyclonal effector T cells could cause could theoretically cause damage to the organ. But I don't see why it wouldn't work.
Yeah, that's very interesting. I'm going to ask if there's any questions from the audience, please drop them in the chat, or feel free to unmute yourself. We'd love to hear from anyone out there. This talk will be recorded so that others can listen to it in future and comment on our YouTube channel as well. And that's always interesting to see what people have to say, you know, when the talks are posted. I wonder, just before we sort of run out of time, what do you think is the biggest sort of hurdle to getting these, you know, this process, this technology aligned with eyelid implantation laboratories? You know, what do you think is the biggest hurdle there? Just getting someone on the project? I mean, is the lab looking for postdocs?
Our lab is, Roncarola lab is definitely looking for postdocs. So yes, if you're interested, if anyone who sees this talk is interested, please go on the Roncarola lab website to Stanford, at Stanford, and apply. You know, maybe the biggest hurdle will be to find people who are experienced with working with human cells, which is, the situation is much improved now than when I was starting my research journey like 15 years ago, when most of the immunology was done in mice, but still, you know, we don't have a lot of people. And we see that when we have postdoctoral fellow applications that know how to work with human cells. It is not the same with humans and mice. Otherwise, it has been done. We had, in the previous institute that I was working, there was an eyelid transplant program, and there was a research lab that was connected to it. So it should be possible. With cell therapies, they are very costly. So that is one thing to keep in mind. But for a disease that is so wide frequent, such as diabetes, the idea of having one-shot costly therapy could be actually cost efficient if the tolerance is achieved, versus, you know, putting the patient on a lifetime of insulin and having, despite of that, having all these side effects that happen with increased blood sugar and inflammation that it induces.
Yeah, I mean, the cost benefit ratio seems pretty clear. If this could be really worked out to, you know, to kind of go work in parallel with eyelid implantation, I think it would be phenomenal. So yeah, if we can get some postdocs experience in working with human cells, you know, we'll provide the information to contact the Roncarola lab, and hope that you know, and hope that some people may find this interesting and reach out to you all.
Yeah, that would be fantastic.
Yeah, it's phenomenal what you're doing. And thank you so much for sharing it. I encourage people to read this paper. Again, that paper we talked about earlier is the alloantigen specific type 1 regulatory T cells suppressed through CTLA4 and PD1 pathways and persist long term in patients. And that came out in Science Translational Medicine, October 27th of 2021. So just a few months back. So it's a really great paper, and I encourage everyone to read it. And thank you again so very much for talking with us today. It's great.
Thank you. It was my pleasure to talk with you.
Thank you.